Good evening, welcome ladies and gentlemen. My name is Luan Ismail, the Executive Director of the ICT for Life Sciences Forum, and welcome to our first event for 2014. We hope to continue to bring you some stimulating and interesting presenters in the course of the year. Before we get on with the formalities, I want to remind you of an announcement we made at the start of the year of a new community partnership, community program. So we have an initiative with Oz Harvest, essentially a gold coin donation we ask for people who attend this event, which goes to essentially provide food for those in need. Every dollar we raise provides 2 meals. We think it's a worthy initiative for the forum to support, and we'd certainly be delighted if you could, on your way out this evening, if you haven't already done so, make a contribution. That'd be wonderful. I'd now like to invite the President of the Australian Academy of Technological Sciences and Engineering, Dr. Alan Finkel, to introduce this evening's presenter. Dr. Finkel.
Look, I'm particularly delighted to introduce tonight's speaker. I get to introduce a lot of speakers, but I don't always know them. Now, I don't know Keith well, but I did meet him 30 years ago, maybe 35 years ago, when my wife was doing a postdoc at UCSF in the lab next door down the corridor. So apart from the particular eminence and striking ability of tonight's speaker, I'm delighted to introduce him on that basis as well. And then when I looked up a little bit about you, Keith, I can see you've not been idle. Back then you were already very well advanced on a course that makes you truly a model for the 21st century scientist. To start, you've stayed active in research, currently working on gene transcription regulation in the laboratory that you still head. But Keith has always adopted another broader role in which he has regularly contributed to public debate on issues into which his training gives him particular insight. For example, in the late 1980s, Keith published on biological warfare. In the early 2000s, the ethics of stem cell research. And he has written numerous other articles on science policy and the role of science in society. More than this, Keith has been active in government and non-government bodies, advocating, advising on, and formulating science policy. So let me give you just a few examples of his long list of activities. Keith chairs the Coalition of Life Sciences. He's on the advisory committee for the Division of Earth and Life Studies at the National Academy of Sciences in America. And he's an elected fellow of the American Academy of Arts and Sciences. He has chaired or served on numerous committees for the National Institutes of Health. He chairs the External Advisory Committee for the Watson School of Biological Sciences. I think that's at Woods Hole, is that correct?
It's at Cold Spring Harbor.
Cold Spring Harbor. And he also sits on the Lawrence Berkeley National Laboratory Advisory Board. Really, I could mention many other similar positions. Back at the University of California in San Francisco, where Keith has remained all these years, my wife and I came back to Australia, but some people are so good the university won't let go. Keith is currently the Vice Chancellor for Research and simultaneously the Executive Vice Dean in the School of Medicine. When we were talking outside, Keith told me that about his engagement with multidisciplinary research and his priority to encourage that and help form alliances between industry, research institutes and the community, all of which is music to my ears. Because like Keith, I believe in the convergence of the medical and life sciences with IT and engineering, coupled with collaboration between universities, research organisations and industry. It's got huge potential. It truly is the way of the future. And I can think of no better person than present this vision for the future to us this evening than Professor Keith Yamamoto. Keith?
Well, it's a pleasure to be here. It's a little daunting to hear that introduction because it does sound like I really have no expertise whatsoever, but write about it stuff anyway. But it's a wonderful pleasure to be here in your beautiful country, and I've had a great time here. Really thank Luan and the forum for hosting me, and so well, and I've learned a lot just in the appointments that I've had while I'm here, and I feel like I'm really gaining not only a lot of knowledge, but a lot of new friends. So I'm very grateful for that indeed. What I want to do is spend a little bit of time talking, and I think it's especially terrific that to come to a place where you can actually drink during your talk. So I'll keep coming back as much as I can. I want to share with you some thoughts and information of activities going on in the United States that I think are interesting, certainly dovetail neatly with some of the convergence research that is going on here in Melbourne and in Victoria, and put a slant on it that I think gives us a compelling reason why we need to be moving toward transdisciplinary research, toward convergence in the life sciences research today. So that's what we'll discuss a bit, is this whole idea of a new look at life sciences research and a new set of new opportunities, really unrestrained opportunities in a time when, of course, all of us are acutely aware that the resources for moving the science are indeed constrained. And I'll say at the very end, I have share some thoughts with you about the importance of scientists being involved in policy matters, in working with policy makers as well as the public at large, and sharing what it is that we do, and hoping to drive in them the kind of excitement that we see as we move ahead in the fields that we all operate in. So what is it that it makes us think that this is a time to really be paying attention to convergence, to bringing together the scientific disciplines? And I would say that the way that I like to think about it is this. I really see science in general, life sciences, at an inflection point. And what I mean by that is that we've enjoyed this fantastic run-up of information to the point that we are kind of, as scientists, surprised ourselves at the level of detail and complexity with which we can describe biological systems, whether it's higher resolution, higher speed, single molecules, single cells, single molecular processes. It's quite amazing. And so we've had this wonderful, thrilling ride of information coming up this slope of conceptual and technical advances that provide really the first detailed description of the remarkable complexity of biology. But I would argue that we are at this inflection point, that we're at a pause, even though we can see on the other side what could lie ahead of another thrill ride. But we don't really know in detail how to get there yet. But what's on the other side is worth it. And that is this, and that is that further advancements in concepts and technologies will provide not only the routes toward detailed description, but really for the first time, the routes to detailed understanding. And you all know as scientists that there's a big difference between description and understanding, a big gap between data and knowledge and understanding. So the question is how we get there. And in a sense, we could sort of underscore the capacity to gather data without understanding by just thinking about what happens if a first-year graduate student in a laboratory comes in and does a gene expression array experiment and looks at every gene that's being expressed at defined levels in a single cell type. And in that experiment is enough data to fill the careers of multiple scientists if they were to chase down every one of those expressed genes, right? But does it give us an understanding of how the genes are expressed, of what that cell is doing, and what makes it the cell that it is? Not really. So you can see that this gap is considerable. So it's here that we've got to worry about what we can do to move forward. How do we move through the inflection point? What's going to be the steps that it's going to take? So I would say there's one, convergence. It's been mentioned already. You know what it is. It is the co-joining of the discipline, concepts, approaches, and really the integration into biology of all these technologies and concepts that have been driving the physical sciences, physics, chemistry, math, computation, engineering, for a long time. And it's those disciplines that will actually drive biology from this descriptive field that we've all enjoyed to a quantitative one. It's not to say that there haven't been people doing quantitative biology for a long time. There have, but but as a field in general, we've gained a lot of description, and we're now having to move into quantitation, and the way to do it is convergence. The second thing is here, and that is to build a new, what I call here a new biology, and I'll explain that term, a new biology ecosystem. One that extends from fundamental studies, basic research stuff, curiosity-driven, non-targeted, out into not only applications that drive our knowledge forward, but to actually be able to take on complex social, urgent social issues. And an important point is this notion that all stakeholders are, all classes of stakeholders are a part of that new biology ecosystem. So what I mean by that is here, this notion of convergence and synergy, that this transdisciplinary science that not only brings together people with different expertise, but knocks down the silo walls between them and allows us to really speak in a common language with each other and move ideas forward in that way. That's what convergence is, that brings the physical and life sciences together. So one plane of integration, if you will, is this notion of building a transdisciplinary language that is common across these disciplines. And while there are pockets of success here again, I would argue that there's no established field yet that is able to really claim that ground. The second plane of integration that's needed is this notion of stakeholder synergy, of taking all of the players that are involved in this game, whether in academia, in industry, in nonprofit organizations, or in the government, and having them build modes of operation and policies that allow us to work together toward common goals. And again, I think we're a long way from that. Even at places where there's been lots of activity of academic industry relations, for example, just to pick a pair of those. I would say that we have a long way to go to making things into a really seamless continuum, and that's really what we're going to need in the future to move forward. So what do I mean by new biology? What is that? What's that all about? Building this common community. So building an ecosystem for life sciences research, and this was the subject matter, the topic header for one of the reports from the National Academy of Sciences that Alan mentioned that I had some involvement in. And here, it's familiar territory from what we've just been talking about. The essence is integration. You merge the subdisciplines of biology, begin to bring them back together, even though we've put a lot of energy into carving out subdisciplines. We can talk about that. The subdisciplinary expertise will still be needed, but they need to be. But each of them needs to be finding a common language to talk together, and then building this working integration across the fields, transdisciplinarity, convergence, and success in that, argued the writers of this report, would create a discovery-driven research engine able to tackle not only really complicated biological problems, but urgent societal issues as well. So that's the imperative that is set up by this concept of new biology, that we can take biological research and move it beyond what we've ever had before of just pushing the boundaries of knowledge in the ways that excite each of us, but also being able to use that information to go after societal issues that actually have no other apparent solutions other than making this work. So I think that's the imperative notion. So what is the new biology in a little bit more detail? In a sense, it is this. It's kind of the primary cartoon from that report. A 4-component cycle that integrates and expands the scope of biological research. So the first of those components starts down here, the separate disciplines. The notion that they would be integrated would be one step. That integration would drive a deeper understanding of biological systems in ways that would allow us to understand organizing principles and move from the capacities merely to analyze to be able to modulate and finally predict. And prediction, in a sense, is the sharpest test of whether we actually begin to understand something. And this deeper understanding, then, would provide the means to use those principles and that knowledge to attack societal problems of real magnitude. And you could see that the committee thought small. They only picked health, environment, energy, and food as a few things to take on. And in turn, the cycle then gets completed because, in a sense, this is like a giant clinical trial. What happens when the clinical trials are completed and a drug is released to the public, then the real experiment ensues. And the same thing would happen here, and that as these things go out into the public domain to carry out these giant scale experiments, that information would feed that deeper understanding, which in turn would feed back on the individual disciplines, and the thing would cycle again. So that's the idea of the new biology, that it would link fundamental discovery to critical societal needs. So the new biology then could, in fact, affect urgent problems. And they are urgent. Over 1 billion undernourished people. That number is still increasing, and the undernourished populations are especially focused in areas where it's already difficult or impossible to grow their own food. Human activities are stressing and altering and destroying ecosystems that we depend on for our not just our environment, but for our health. The transportation fuels still depend almost fully on non-renewable resources, and this healthcare decisions, as we all know, are still based on statistics, rely on costly technologies that may or may not benefit individuals, but they get deployed anyway, driving up the cost of healthcare in unsustainable ways. What's worse, or what's really apparent and is something that needs to be kept in mind, is that while each of those problems of food, energy, environment, and health is daunting and right now has no real solutions, the fact is that they can't be solved individually. They all have to be solved together. This is an integrated problem. So the food challenge, and it's a big one, 50% increase in food productivity by 2050, and there's really no way that we have with current technologies to achieve that. It must be met without increasing land and water use, fertilizer and pesticide release, or fossil fuel input, because you know what those will do to the environment and what destroying the environment will do to health and so forth. So we take something we already don't know how to do and we put constraints on it that says, oh, by the way, you have to do it without using any more water, land, or dumping more stuff into the environment. Meeting the health challenge depends upon sustainable, nutritious food sources, of course, and maintaining ecosystem services, both of which are under great threat. And meeting the energy challenge with biofuel production is no fair planting everything to corn that can be turned into ethanol, and then people starve because you stop growing food. So you get the picture. It's an integrated set of problems that can't be solved individually. Now the good news is that while they're all tied together, so are the solutions. The problems are tied together, but so are the solutions, really because there's one biology. So a deeper understanding of microbial communities, for example, would better define plant ecosystems, human immune responses, help to optimize food crop growth, because we would understand the relationship of the plant microbiomes to the plant's nutrition and growth, would help to identify biocatalysts for biofuel identification, and so forth. So understanding the microbiome and microbial physiology and expression patterns would help in each of the areas. This one's even more obvious and in a way relevant to some of the stuff that I'll have to say later, and that is handling data. Massive data sets we're immersed in them every day. Massive data set handling, high throughput imaging are essential for ecosystem monitors, for metabolome analysis in human health, for manipulating plant stress responsiveness, for thinking about how to grow plants under what are now stressful environments that don't allow them to thrive. So that's the good news, and that is that because there's one biology, if we can solve one problem and we talk to each other, then the problems can actually spread across other areas, other domains as well. So why aren't things just cranking along full speed? And it's because there are barriers to success, and this is just a few of them. In the US, federal support for life sciences is fragmented across 24 federal agencies. Federal agencies that have a stake in not talking to each other, because they've got to go defend their budgets every year on Capitol Hill, and one of the ways they do that is by saying, if you shut off our money, this crucial stuff will come to a dead halt because nobody else is doing it, because we're not telling them what we're doing. Databases don't talk to each other. Just one of those 24 federal agencies, of course, is the National Institutes of Health, made up of 27 sub-agencies that don't talk to each other. Rates of incremental advances are insufficient to be able to get us there. So simply allowing scientists to work in areas of their curiosity without enunciating some vision that will begin to move people forward in a particular direction is a problem. We'll get back to that. The capacity for data collection, manipulation underscore the need for advances in quantitation and interpretation. So this is the moving from description to quantitation. I'll underscore that in several places. Here's one. Life science education underemphasizes quantitative studies. So what's needed? Coordinated funding, maybe enunciation of core challenges. We're going to get back to that in several different ways as we go forward, but you can see that one of them is a report like the new biology report that tries to coalesce a set of ideas to move people, funding agencies, scientists in particular directions. And we can talk about whether there are weak spots in that kind of approach. Advanced technologies, computation, quantitative education. So what was called for in the report then was a national new biology initiative, right? So an enunciation of a grand challenge that says we have to pay attention, we have to put some focus on moving in particular directions. An initiative that would move agencies that normally would never talk to each other to work together to fund projects of common interest across agency boundaries that are clearly multi-transdisciplinary and that are long-term. We, like you, are saddled with federal budgets that go year to year, and you all know that your projects don't go year to year. The missions then would be to promote bold cooperative approaches to understanding complex biological problems and to define and achieve solutions to societal challenges across that sector of 4 big problems. And there are cross-cutting priorities as well. We've talked about them. The imperative to develop information sciences and technologies and to develop interdisciplinary, transdisciplinary curricula at all levels. So why a national initiative then? Because big goals capture imaginations. The enunciation, a clear enunciation of a big challenge itself will drive toward success. And we saw that in the United States in the 1960s when Kennedy said we're going to go to the moon in this decade, and they made it in '69. At the time he enunciated that challenge, we didn't really know how to get rockets off the ground that well, much less take a man to the moon. When the Human Genome Project was adopted in 1989, it was clear that we had no way to sequence the human genome, right? People were still making sure that their favorite Maxim Gilbert gel plates didn't break. So we're sort of a long way from the human genome. we need the expertise of many federal agencies, scientific communities, academic and private sectors to come together to work on these goals that have this kind of dimension to them. Because the problems that are inherent in solving, in addressing those problems are far greater than what an individual science or an individual scientist or an individual discipline can provide, because you quickly run into other issues that need to be solved in order to get there. Funding is only one of them. And this great need for multi-agency collaboration and leadership to define the best investments in foundational sciences and technologies and advance this interdisciplinary, transdisciplinary education. So let me just give you a couple of examples drawn from 2 of those 4 grand challenge areas. A quick look in at a couple of different approaches to the food challenge, and then a more extended, actually almost the rest of the talk, looking at one health challenge and one approach to the health challenge. So here is a new approach to a new biology food challenge. Here's a vision, and that is, what if we could adopt any crop plant to any growing condition? Right? So you want to grow twice as much food. Maybe if you can grow peaches in the desert, you're going to be in good shape. I don't know how good peaches are to take care of the food problem, but the mission then would be a sustainable increase in local food production, food that can be grown in places right now that's difficult, and you don't have to grow everything in Iowa and ship it with gasoline around the world. What is needed then is understanding, information about genetically informed breeding of transgenics, recognition of the importance of biodiversity, systematics, evolutionary genomics, analysis of crops as ecosystems, looking at influences of the microbiome and the way that a crop plant grows, for example. This requires new understanding, plant development, growth, and productivity, plant tolerance to extreme conditions, plant adaptation to climate change. When I was in a conference where we talked about this idea of adopting plants to extreme growth conditions, and I gave people the example of cracks in my driveway, which plants always grow out of. I don't water the cracks in my driveway. The soil in the cracks in my driveway isn't soil, it's just sandy grit. It gets engine oil dripped onto it. You put plant poison in there, plants grow out of the cracks. Plants know how to do this stuff, right? If we could teach plants that we care about to do that stuff, that would be cool. Here's another one, and this came out of a workshop that was sponsored by the National Institute of Food and Agriculture from the USDA, U.S. Department of Agriculture, Department of Energy, Office of Science, and the Gordon and Betty Moore Foundation, an aggregation of 2 federal agencies and a private foundation, and the Howard Hughes Medical Institute. The vision was to achieve carbon neutrality in the agriculture and biofuel sectors, and a bunch of deliverables were generated out of this workshop that were set up sort of as challenges, and you can kind of scan the list and see, and they are indeed challenging. Understanding carbon flow and defining its movement through production and use systems, developing biofuel feedstocks that prosper in local environments that are not suitable for food production, and so forth. So there's activity, there's movement in that area, that challenges have been set up, there are meetings that are taking place, and some of these challenges are being enunciated in ways that we hope can capture people's imagination. I'm really going to spend almost the rest of the time on a health challenge, because it gets back to this imperative for convergence on the one hand, and communication to policymakers and the public about what it is that we do on the other, involving the public as kind of owners of a research process that we're a part of as well. And that's here. The vision is health and health care tailored to each individual rather than being driven by statistics. And the mission is to create this concept of precision medicine, which I'll define in a moment. What's needed? Integration and analysis of vast biomedical data sets, understanding of microbial, environmental, experiential factors, kind of the soft and fuzzy stuff that we don't really get yet, how to be able to quantitate and understand, and synergy between scientists, clinicians, and citizens. So we talk about building good relationships between the basic scientists and the docs, and we all of us who have medical or affiliated with medical schools has some flags, victory flags to wave about stories that we can tell about some fantastic things that have happened in our institutions. But in general, those fantastic things are driven by the smart individuals that we're able to hire into our institutions, and not by things that the institutions do, policy-wise, that really move things together and take down the barriers. And the citizens and the patients, right, they're consumers, customers, and not part of the process. And the precision medicine concept argues that that needs to change. We need integration across that sector. It will require, this creation of precision medicine, will require a fundamental understanding of lots of things. Here are 3 examples: pathways of growth, development, and differentiation, external influences on physiological processes, things like environment and experience, and therapeutic design that is rational, understanding the mechanisms by which therapeutics work, and what happens with what always happens with drugs, and that is resistance. So where are we now? How far along the track are we to creating precision medicine? And I would argue, as you see here, that we're practicing imprecise medicine. And the public can lay this out as well as anybody, because they open the paper every day and see that some new disease gene has been discovered. And so it will be fair for them to say, what the heck is a disease gene? Because a disease gene doesn't mean that you have the disease. If I have one, why does it only change the risk, sometimes by tiny amounts, of my getting the disease, right? People freak out when they read the lead paragraph that says, if you have this gene, your risk for lung disease goes up 5-fold. And they get down to the last paragraph and it says the 5-fold is from 0.02 to 0.1. Why is disease diagnosis so far from an exact science? How could gathering information about me and comparing it to information about many others move me actually, toward a new level of understanding of disease and toward precise diagnosis, treatment, and cure? Pretty good questions. And the person on the street, there are many people on the street that could ask questions like this every time they pick up the paper. Well, here's some answers. Here's some answers to those questions, not the solutions to them. And really the main answer is what we've been discovering over the past 25 or 30 years, and that is that normal biology is complex in ways we didn't anticipate. It's really complicated. So normal biology is complicated. We don't understand mechanisms by which the processes work. We know that we appreciate in more detail every day that they're controlled not by pathways, but by vast networks interacting in ways like this, right? Through big signaling networks, not linear pathways. That they are highly context dependent, moving from individual to individual or cell type to cell type, or one temperature to another, one physiological state to another, one age to another, and so forth, altered by many, many variables. So if normal biology is complicated in this way, now you can rationalize, can't understand, but you can rationalize why disease is complicated, because it's just an aberration of normal biology. So it's going to be complicated. Right? So that may not make the person on the street feel any better, certainly if they have a disease, but at least they can maybe have a little bit more appreciation or understanding of why the complexities are there. So if that's imprecise medicine, what can we do about it? And what we argue in another report that I'll describe to you from the National Academy of Science, in the U.S. National Academy of Sciences, is precision medicine. So now it's fair for you to ask, what the heck is precision medicine? And I think one way to carve it out is to kind of corner it and say what it is and what it's not. Let me start by saying what happens in current medical practice. In current medical practice, you go into your doctor's office and you say, Doc, I've got this deep pain in my shoulder. And so the doctor says, okay, cool, that's really too bad. I'm sorry about that. Let's take your vital signs. So they measure your blood pressure and weight, height, weight, and so forth. And she goes over, hopefully, to her computer, not to a file cabinet, but maybe to a file cabinet, and looks up the last time you showed up in her office. And says, first of all, it's been 15 months, where in the heck have you been? And looks up the vital signs measured then, and if you had any complaints about your shoulder, you didn't. And makes, based on her expertise as a doctor and stuff that she's seen before, she makes a diagnosis and maybe writes a prescription. And thank you very much. Your 15 minutes is up. Now, what happens in personalized medicine? Personalized medicine, your doctor says, boy, you're lucky you came to me, because we do personalized medicine. And we are not going to be satisfied with your vital signs and what happened 15 months ago and what you tell me today. And we're going to collect, maybe we have already been doing it, collecting a lot more information about you, right? If you're at the University of Melbourne hospitals, maybe they've been doing this periodically. You've been showing up every few months to get provide some kinds of data about yourself, right? By giving blood or sweat or something. And they're analyzing stuff, gathering lots of information. Maybe your genome is being sequenced or has been sequenced. And a whole bunch of data then is collected about you, and your physician can now make a much better diagnosis and treatment plan than she could before. And she says, fantastic, this is much better, and here's your prescription, and here's what you have, and thank you very much. So that's a fantasy, right? We're not there yet. Right? We're getting there, but we're not there yet. So if that's personalized medicine, what the heck is precision medicine? Precision medicine says, boy, are you lucky you showed up in my office, because we're doing precision medicine. And we don't depend on your vital signs from 15 months ago and today. We don't depend on your your genome, right, and your behavior and what you were exposed to between ages of 8 and 15 growing up in a pollution-ridden city somewhere. But we're going to use data from everything and everybody and aggregate that in a vast computer system that is able to be predictive in ways that no amount of information about you alone would do. So a massive data network that aggregates and analyzes information from a huge patient cohorts, not just you, but huge numbers of people, maybe millions of people, tens of millions of people. And not just sick people, but healthy people. And not just people, but fruit flies and worms and yeast and bacteria and mice and so forth, all using these convergence-based approaches, attacking the problems using this transdisciplinary set of approaches to reach toward an understanding of disease mechanisms and toward a precise diagnosis and treatment plan that is tailored for you based on this massive amount of information. You can see that each of those three is focused on you, making a diagnosis for you. But it moves from depending on anecdotal data to more detailed information about you, to information about all biological systems that can then be predictive for you. So it's a big difference. Here's where we are. We're not here yet, and we're certainly not there yet. And we would argue that this report I'm going to show you argues that that's where we need to go. So precision medicine then is really about building this biomedical and health continuum. It says it's important to be able to integrate this vast and expanding range of information about biological, behavioral, and experience, to drive basic discovery with new syntheses and approaches. So this feeds new ideas to basic scientists, to promote the development of a more accurate and precise classification of disease. I'm going to get back to telling you why that's important in a second, and to advance the speed and accuracy of diagnosis, therapy, and cure. So that's a grand set of challenges. How do we get there? And so this report from the National Academy of Sciences says, build a tool that does it for you. So this report, which came out at the end of 2011, it's called Toward Precision Medicine, Building a Knowledge Network, keyword, for biomedical research and a new taxonomy of disease. There's a URL that can allow you to download the report for free. And what's the enabling tool? The enabling tool, and again I'll define these terms, is to build a computational information commons that brings together all biological information, not just about tens of millions of people, but about other stuff. Matures that information into a knowledge network, and that is able to correlate and align disparate information, motivate research team building across disciplines, of course, and to convene all stakeholders, including patients, into this dynamic research ecosystem. That sounds good. So what's the problem with disease classification? And that we need to change that in order to get to the rest of the stuff. Right? So we would argue, this report argues, that disease classification right now is a barrier to progress. It's because it's descriptive. Disease classification, as you know, is based primarily on organs and symptoms. Right? And that means that the people that are studying a different organ and a different symptom don't talk to each other. It's inflexible. It creates rigid silos in research, education, and funding. Medical schools are separated in departments of organs and symptoms. The U.S. National Institutes of Health is separated in institutes of organs and symptoms. And there's no real place for basic science in this. It's narrow. There's no natural fit for fundamental discovery research, and no incentive to build the continuum that we're talking about is essential. In fact, there's sort of an anti-incentive. It's better to keep information to yourself. So that's not good. So what could we do that would be better? What if we classified diseases instead of organs and symptoms by mechanisms, the things that actually lead to the disease? Well, again, the person on the street actually knows that that wouldn't be such a bad idea, because the person on the street knows that there's at least two kinds of diabetes. They probably don't know that type 1 and type 2 themselves have multiple mechanisms that cause them, but they do. The person on the street knows that there's not one kind of breast cancer, there's a bunch, and that they've kind of heard that that's true with cancer in general. It's a constellation of diseases that have different causes. So why are we just throwing them together and treating them like one disease? So I want to spend a couple minutes talking with you about breast cancer. Because in a sense, the first really personalized medicine or move toward precision medicine came from a development of a new drug that would treat a specific version of breast cancer, and that was breast cancer that led from the overexpression of this gene called HER2. So that's one kind of breast cancer, is women who overexpress the HER2 gene. So let's look at that. So HER2 is a disease gene of the kind of the front page news variety. HER2 overexpression, though, does not predict breast cancer. Rather, it predicts increased risk for breast cancer. Breast cancer can occur without HER2 overexpression. So having HER2 overexpression does, in fact, having HER2 overexpression doesn't even mean that you have breast cancer. It means your risk goes up, right? And if you have breast cancer, it doesn't mean you have HER2 overexpression. But Herceptin works on every breast cancer patient who overexpresses HER2. So it's precise in that sense. If you're a woman who has breast cancer and is overexpressing the HER2 gene, this drug will help you. All right, so it's precise in that sense. But Herceptin doesn't cure breast cancer. The patients all die. Their life expectancy goes up by a year, right? That's important if you have breast cancer. But it doesn't cure the breast cancer. So what we can say, now you can say this, is that HER2 overexpression provides a mechanistic classification of breast cancer. Overexpression of that gene, right, will be helped by Herceptin. But because disease is complex, Herceptin alone is not a cure, even for that specific subclass of breast cancer. Maybe as studies go forward in understanding mechanisms, we'll be able to stratify that class again of HER2 overexpressors, so we know ways that there may be a subpopulation that could be helped longer, or we may find the pathways that go around the Herceptin treatment and be able to go after those. Right now, we don't know. And so it gives us a way to classify the breast cancer better in ways that are therapeutically efficacious, but Herceptin alone is not a cure because disease is complex. Okay? So we're back to this notion of why it's good to classify disease by a mechanism. And here's something that a person on the street and many scientists actually don't know or don't appreciate. And that is, not only is it true that diseases have multiple mechanisms, but it's also true that mechanisms cause diseases that don't look like each other. Right? My favorite example are the constellation of diseases that are caused by cilia defects. Cilia are a little hair that go off of each of your cells. Right? And there's a pathway or a circuit that's involved in making the cilium. And defects in making the cilium, or making it correctly or expressing enough of it, lead to situs inversus, a fascinating disease in which your organs are on the wrong side of your body. Polycystic kidney disease, water on the brain, retinal degeneration, polydactyly, too many fingers. Those diseases don't seem like they're so similar, and the people who work on them are not talking to the other guys. They're all caused by the same defect. Doesn't seem so good because they're not talking to each other now. So a mechanistic classification of disease could capture and use more information for research than we're currently capturing and using by the current classification of disease. So the current classification of disease is a problem. It's a barrier. It's getting in the way, keeping people apart that should be talking to each other, probably keeping funding agencies apart that should be working together. And could use this information for better research, education, and healthcare. So that green line looks good, but how do you do it? You don't do it by making a department of cilia, right? Nothing would be cilia. So that's not going to work. So what do you do? So now we're back to precision medicine. So this is now the kind of primary cartoon from that report. And I'm not going to go through it in any detail. I'm just going to give you kind of the gestalt. And that is this. This report said, instead of making a department of cilia, make an information commons that's able to reach into existing databases and pull that information in ways that it can be analyzed. These very disparate pieces of data, different kinds of databases, different kinds of information, brain imaging and genome sequence, and small molecules in the blood or sweat, and people's experience, and mount it on a platform so the information can be compared and analyzed. And those comparisons and analyses will reveal correlations and connections that will drive the formation of this thing called a knowledge network. And that's what's cartooned in the spaghetti up here, and we're going to look at that in more detail in a moment. But the key thing is that this information commons leads to correlations that make a knowledge network, that make new knowledge come out, spill out the bottom. And that new knowledge is substrate for basic scientists that make new hypotheses and design new experiments. And it's substrate for clinicians that look for new treatment paradigms or diagnostic approaches that lead to clinical trials, that lead to new information on the clinical side. And the information that comes out of those new basic science experiments and clinical trials spills back into the information commons, builds it larger, builds more knowledge, more connections that can be made on the knowledge network, and better knowledge spills out the bottom when you do it again. So in a sense, really all this does is it says, let's find a computational way that will drive all of the information together. And of course, what that does is it drives all the people together and makes the stuff that gets discovered in a basic science laboratory, even by basic scientists that will yell loud and put their fists on the table that they don't care about disease. It makes it relevant anyway, because it's all being homed in the same place and can be used by the people who do care about disease. So here's the drill. This is basically the idea, which is just a direct lift from Google Maps. So you know the way Google Maps work. It takes different kinds of information, right, and stacks all those information on top of each other. And in the case of Google Maps, aligns all the information by geography. So you could take a plumb line and drop it straight through each of those layers, and you would know, right, for a given transportation system, other ways that the land is being used, where you are in the census tracts, what buildings are around in that area, just by dropping that line straight through and being able to link together those different kinds of information. So the idea of the information commons and knowledge network is exactly the same. Stack up these very different kinds of information. But in this case, it's not oriented. We don't know enough to orient it. There's no common point like geography. And so instead, you have to work to figure out where the connections are. That's research, right? But the tool is stacking up the information in ways that you can use it for analysis. So that's the drill, to stack up those different kinds of information, all the way from the genome sequence to the individual patient medical record and stuff in between that goes beyond the kind of objective comfort of omics to kind of fuzzy experiential stuff, right? Every molecular biologist knows that the environment and experience affects the risk for disease, whatever that means, but they don't want to talk about it because we don't have ways to quantitate it and organize it and compare it to the genome sequence. And so it's just like, thank you very much for telling us that. Please go away. We're doing science. And this says we can't do that anymore. There's too much information there we need to understand. And so we got to use it all. And now you begin to build interlayer edges between those nodes in the different layers and see what the correlations tell you or suggest as hypotheses. We start out with a lot because the objective omics stuff can be linked together pretty well, and pretty quickly you get to some pretty lower densities of correlations and less things you have less confidence in, but you build them anyway. And the network grow and change as information is added. So if you know that this spot in the proteome is due to some variation in the environment, further research may reveal that that spot in the proteome is also due to a component of the microbiome. And so at least the basic science question arises, is this component in the microbiome what is the variable in the environment that's causing this spot to come up, and so forth, right? So there's a basic science hypothesis. So networks grow and change as information is added. So if this information commons and knowledge network works, then it would serve three key purposes. It would enable new basic science discovery. We just talked about an example. It would improve clinical care because of the information that's feeding back up in this direction. And it would inform the taxonomy, the classification of disease. Maybe we would understand why Herceptin treatment doesn't cure HER2 overexpression breast cancer. So how do we use this? So imagine that you could actually see it on your computer. See, clouds, it wouldn't look anything like this, but smart computer data visualization people would allow you to turn on your computer and see a big cloud of information. And so you could put this advanced visualization would allow you to sort the information in lots of different ways. You could click on a node or an edge or sweep out a zone and collect information and define not only an idea, but a potential team of coworkers who think about things differently than you, but in fact are thinking in the same area, using very different approaches. And if you work together, maybe you could ask and answer questions that none of you could ask or answer individually. The nodes or edges then are populated with investigators. That's the key. And so this happens to scientists all the time. You come across your work carries you into a new area that you don't know anything about. And now you can find out quickly who else is working in that area, who knows about a lot about it. And if you could work together, you could go do something that you couldn't otherwise do. And you get to design the team. The teams would be designed self-assembled with the goal of establishing new nodes and edges, maturing the knowledge network in ways that are informative to all members of the team. So the knowledge network then would drive transdisciplinary research. It would drive convergence in ways that is based solely on being able to capitalize upon knowledge to do some fascinating new things that you couldn't think of before. I'm not going to go through all of these, but they're nice features. It's an enabling tool, not a directive one. It's not the dean telling you to work together with Joe Blow down the hall. The teams are self-assembled, they're flexible, they're dynamic, they can go away when the experiment doesn't work or it works great and you're done with, the team is done with a molecular biologist and needs an epidemiologist. It's scalable, it can be done locally or go to the federal level or go to the multinational level. And it impacts all stakeholders. Notably not just academia and industry and so forth, but in fact, it could impact the way the public thinks about science, and that's important. The public picks up the papers every day, sees the new disease gene report, and sees that the government agency is now giving different advice very firmly than it gave very firmly a year ago. And they're saying, what the heck's going on? Are these guys flipping coins somewhere, or what's happening? We're giving them money to do this? And if they can understand that it's because of maturation, new knowledge that have built a new edge between nodes in a knowledge network, that it drives our thinking in a different way, then they could see those changes as victories for scientific research and might think differently about the way that they support science and scientific research than before. So at UCSF, we're doing a pilot experiment. We're trying to build what we hope will be a national and international model. Just within UCSF, and I don't mean by that we're controlling every step at all. We're doing this with many, many different partners, but trying to build a little model at the institutional level. The goals are obvious and clear. That is to be able to use information, the flow of knowledge that comes out of the knowledge network and other elements in this thing we're calling the precision medicine platform. And to be able to use information that's incoming from patients in ways that feed stuff back into the research process and to the patients and citizens themselves. Here are the components of the knowledge network. I'm not going to go through them in any detail. I'm going to flash through an individual slide for each of the components without dwelling on them. But if you look at these, omics, medicine, genomics and proteomics and other omics, a computational health science institute, digital health, something that's an important revolution that is coming to us, all feeders into basic and clinical discovery, along with data coming in from patients and citizens, all of that flowing into the information commons and knowledge network and dropping new knowledge out the bottom. So as I said, I'll flash through a slide for each of these, probably highlighting maybe one feature of it as we see, but we've built a committee that is populated with leaders in each of those 6 areas. And the committee, because the areas overlap, we meet a lot to talk about what's going on and what we could be, making sure we don't miss stuff that falls in between. So basic discovery, if you scan the white parts of this, you see are just kind of, in a way, a description of the basic research process, but it's a part of the platform, and so the information is all flowing into the network, including establishing these new connections between layers using convergence approaches to build a common language. Clinical discovery is normal clinical research and clinical trials and so forth that you're familiar with, but includes a big set of charges for privacy and security, regulation, and so forth that are going to be critical and become just more critical as you start thinking about the daunting challenges that arise from having vast amounts of information that are tied to individuals. Omic medicine, the collection and analysis of human subject and other information in a validated, government-certified environment. A lot of challenges that are associated with that. We're beginning with genomics like you are and many other places are, but we have it in mind that we need to be able to extend what we learn and approaches that we can establish out to the other omics as well. And with that comes really an imperative to build educational programs that will bring together the communities that are able to capture and then use this information. Computational health sciences, this is going to be a driver for allowing the knowledge network to be built. It's essential, and we're bringing in card-carrying computer scientists to think about how to code this information. And the Center for Digital Health is a revolution that is coming at us at full speed, that the public expects and wants increasingly. Passive sensors and reporters of information about themselves and their own health data that they can use for themselves, and hopefully will be eager to be able to share with units that are building this precision medicine knowledge network. As I've said, if we don't, in our profession, don't respond to this, we're going to get run over from behind by our patients. And already we're facing this with people coming in with weak information drawn from the internet. And then the knowledge network. We've chosen to nucleate ours at UCSF in two big sprawling disease areas, neurological disease and cancer. But of course they merge as the network begins to get built, and that's beginning now to happen. So that's really the basis of the pilot study, is to do this in ways that serve the clinical and basic research community and the public and patients, sick people and well people that want to keep it that way. So precision medicine then is this response to a new biology health challenge. It's a convergence implementation that motivates integration and advances research and health. It builds a research continuum driven by an information commons and knowledge network. It is a data-driven platform to maintain health and to diagnose, treat, and cure disease, and the potential outcomes make the effort and challenge worth taking on. Building a research ecosystem that informs, integrates basic through clinical, includes all stakeholder sectors, health and healthcare tailored to the individual. And I haven't talked about this, but we're convinced that this would actually drive healthcare costs down, and we're on a worldwide basis all on an unsustainable track. And something needs to be done about healthcare costs, and we can talk about that if you are interested. So we started with this idea that we're at this fantastic point in doing science. It's a great time to be doing science, and to be sitting at this inflection point, thinking about how to get through the set of challenges, because we know the rewards on the other side are so good. And so I want to end with just this notion, and that is that I've talked about three grand challenges that have been laid out in the U.S. biomedical research community. New biology, precision medicine. I haven't talked about this one at all, but you're all aware of the Obama Brain initiative that is just beginning to go forward. And I think that these are a legitimate set of questions to ask about this strategy and whether it's something that we should be embracing as we move forward, try to move forward in our own areas of interest. Will grand challenges capture public imagination of policy makers? It's in a way the only rationale for them, and because it garners public support and policymaker support that should turn into support for the kind of work that we do. A question that has to be asked probably first. Will they stimulate multi-agency coordination and support? Will they enable or inhibit investigator-initiated research. The good ones, we would argue, will always enable that kind of work, allow any individual scientist to look at a great big area of grand challenge and say, you know, I can make a contribution over here. So I'm going to go in for this money because I can do something that will move the ball. And will they be embraced not just as U.S. national grand challenges, but things that involve us all. We think that this is beginning to happen. In 2012, April 2012, President Obama released something called the National Bioeconomy Blueprint. And if you look at that document compared to the New Biology for the 21st Century, you will see that a clever person named Mary Maxson at the Office of Science and Technology Policy took this report and said, boy, the only way we're going to get support for this is if we rewrite it in economese. And so the report is a direct translation into a different language called Make the Economy Better. Right? And then Obama announced it as the National Bioeconomy Blueprint. There's a bunch of precision medicine activities going on. I'm not going to go through them in any detail, except we were gratified last week when Jerry Brown gave his 2014 State of California address. And he said, just as California led the way with stem cell research, so too can we pioneer the new field of precision medicine, which uses genomics, medical devices, computer sciences, and other fields to treat individual patients instead of broad populations. Man, that's what we want. We don't have the governor of California running around saying this stuff. Some of you know that I've had some criticisms of state governments getting involved in biomedical research. We're hoping that this can turn into some resources and public support that'll end up making a difference. And Obama has announced his BRAIN initiative, which right now is all potential. The first grant just got given last week, and we'll see if it does some of the things that I think grant challenges need to do. So I think that this is a legitimate set of questions to ask ourselves whether this is the kind of thing we need to be doing. But the important part of all of this is that in Australia, in the United States, there's no doubt that life science research is a vast public trust. Right? In the US, it's really no fooling. That in the U.S., citizens provided about $90 per capita to support NIH research, about 1% of the federal budget, right? So we complain about not having enough money, but that not enough money is a lot of money, and so from it comes some responsibilities. We know that life science research has advanced spectacularly. You've been a part of that. Making that happen, and now has the potential to move things forward to address urgent societal issues. But with this kind of public support and public trust, then, comes a responsibility for all of us to communicate with and educate citizens and policymakers about what it is that we do, about scientific discovery and its potential applications that I think we're now really on the cusp of, and something that we need to be not only excited about with each other, but talking about other people that can benefit from it and make things happen. So let me stop there. Thank you for your attention. I'd be happy to take questions.
Will you join with me in again thanking Dr. Keith Yamamoto for his very excellent, inspiring talk. Thanks very much, Keith. Thank you. We look forward to seeing you at the next ICT for Life Sciences Forum presentation. Thank you and good evening.